Thank you for the opportunity to present. My name is Joe Rozowsky, and I'm presenting on behalf of the EN-TEx working group, and I'm going to tell you about the ENCODE EN-TEx resource. EN-TEx is a project using samples that were sourced from a collaboration between the ENCODE consortium and the GTEx consortium. And the main goal is to catalog genomic elements in personal genomes from individual sourced from GTEx, where we have matching ENCODE datasets, functional genomic datasets, across a breadth of assays and tissues on individuals where we have the matching corresponding personal genome sequences. So, as I said, we've got the matching whole genome sequences and functional genomic data assays for assays such as RNA-seq, ChIP-seq, ATAC-seq, HiC, and so on, across approximately 25 different tissues for 4 different individuals. And in total, this encompasses about 1,500 data sets. And all these data sets are fully consented and open access and available from the ENCODE project portal. And the goal of this project, which we're in the process of wrapping up, is to catalog all the genomic elements across these individuals and different tissues, and to study the effects of variation within the different genomes in terms of how they affect these genomic elements, and to fully investigate the importance of using the actual personal genome, diploid genome sequences for these individuals in analyzing the functional genomics data coming from these specific. This slide shows the data sets. So as I said, there's about 1,500 total data sets from 25 different tissues, and the tissues are listed as the column on the plot. And then the rows on the plot correspond to all the different ENCODE functional genomic assays that have been performed on all these data sets. So as you'd expect, there's the genome sequencing, there's various forms of ChIP-seq in terms of TF-ChIP-seq, histone modifications, other things like DNAs and ATAC-seq, HiC, and a variety of sort of RNA sequencing experiments in addition. And the way this sort of plot is shown is that for the 4 individuals, it forms a square, and if all the data sets are present for the 4 individuals, all the 4 shades are shown, and there's 2 males and 2 females. As I said, all this data is available at the Stanford ENCODE. Just as a vignette to show the utility of this data, one thing we've investigated quite thoroughly is allele-specific activity. An example of which is, say, allele-specific binding, which you could measure using ChIP-seq data. And the idea is if you've got a transcription factor binding site that overlaps a heterozygous variant, you can look at the sequence data, the sequence reads, and differentiate reads that arise from one allele versus the other allele. And in the case of a binding site, which the majority are balanced in terms of the binding being equal to both alleles, a subset of the binding sites might only come from one allele versus the other allele, and we could use the differentiation between the reads mapping to both alleles to determine instances of allele-specific binding using statistical tests. One could do this beyond allele-specific binding for the majority of the assays we have in terms of RNA-seq for transcription, HiC, and so on. And beyond this, in terms of looking at the allele-specific binding at the heterozygous variant level, we can aggregate into sort of broader elements. So for ChIP-seq, it would be transcription factor binding regions, or in terms of transcription, it would be like transcribed regions such as genes. And we could also aggregate allele-specific behavior by combining different tissues or across different individuals, or a combination of tissues. And so as part of this project, we've constructed a comprehensive catalog of allele-specific elements in all the different assays, separated out either by individual tissue or sort of some combination. And this just sort of is an example to illustrate the power of allele-specific binding. This is a known imprinted loci, H19 IGF2. where there's a differential behaving between known differential behavior between the maternal and the paternal allele, where H19 is expressed on the maternal allele, but only IGF-2 is expressed on the paternal allele, and there's a CDF silencer that is differentially bound between the two alleles and sort of inhibits the expression of the IGF-2 on the one allele versus the other. And when we look across the breadth of assays that we have, we could actually see exactly this imprinted behavior by looking at a combination of the RNA-seq, the CTS ChIP-seq, and other data sets. And we could see this in this behavior in a number of different tissues. So just in summary, this resource, and we're in the process of putting together a paper sort of describing a summary of these results, many more things that I didn't have the time to talk about. And this data, I'm not going to go through all the labs, but there's a number of people, a number of different labs within ENCODE that contributed in terms of the data production and the data analysis. Thank you.
Hi everyone, today I'll be talking about the transcription factor regulation of genetic variant effects. So when I say genetic variant effects, I'm talking about EQTLs, or expression quantitative trait loci. And you can think of these as variants that are associated with changes in gene expression levels in a population. We know that EQTLs are enriched in cis-regulatory elements, and many co-localize with GWAS loci, which suggests that many EQTLs exert their effects via non-coding mechanisms to affect human health and disease. EQTLs can be context-specific, such as in the left, where an EQTL is present under immune stimulation and not at baseline. And EQTLs can also vary in their effect size across contexts, such as on the right, where an EQTL may be present in multiple tissues, but with different effect sizes. We know that EQTLs are enriched in transcription factor binding sites, and this makes sense as transcription factors are one of the few sequence-specific interactions with the genome that could be directly affected by a genetic variant. So here on the right, the transcription factor has a low affinity for this A allele, which results in a low presence of the gene expression. And on the right, the transcription factor has a higher affinity for the C allele, which results in high levels of gene expression. We can think about this relationship at different transcription factor levels. So at a low transcription factor level, we might not have binding of the transcription factor to either allele, which will lead to low gene expression of both alleles. And at very high transcription factor levels, we might saturate binding to both of the alleles and have very high expression for both alleles. So in this scenario, in the middle, we would observe an EQTL effect, as there's a difference in gene expression for the two alleles. But on this situation on the right and on the left, we would not observe an EQTL because there is no difference in gene expression between the two alleles. So to investigate this transcription factor mechanism, I'm using 3 data sets. The GTEx V8 dataset contains 17,000 postmortem tissue samples from 54 tissues of 838 donors. We have RNA-seq data from every sample, as well as whole genome sequencing of every individual. I'm also using ENCODE transcription factor ChIP-seq peaks for 169 transcription factors, and HOCO-MOCO consensus sequences for 169 transcription factors. So from the GTEx data, we selected the variants that were possibly causing an effect in gene expression changes, so based on fine mapping of EQTL in any tissue, and variants that overlapped a transcription factor ChIP-seq peak as well as overlapped a motif. So for these about 500,000 variants, we asked, do we see a relationship between transcription factor levels across tissues and EQTL effect sizes across tissues? So here, each point is a tissue, and you can see that at lower transcription factor levels, we have smaller EQTL effect sizes than at higher transcription factor levels. We also asked, within a certain tissue, do we see a relationship across individuals between transcription factor level and EQTL effect size? So here on the left, we do see an EQTL effect in adipose tissue as there's different gene expression for the 3 different genotypes. And we also see an interaction with the transcription factor level. However, for skin tissue over here, again, we see an EQTL effect, but we see no transcription factor effect on that EQTL effect. So here we would say that across tissues, we see a transcription factor EQTL relationship, and within adipose tissue, we see a transcription factor EQTL relationship. But in this skin tissue, we do not. So I did this for all the variants, and across tissues, we found about 400,000 relationships between a transcription factor and an EQTL effect size. And we found in different tissues, differing numbers of interactions from 50 up until about 10,000. Next, we asked, how do these two data sets relate? And we found that we have about 10,000 transcription factor EQTL pairs that have at least 2 lines of evidence that that transcription factor interacts with that EQTL. So next we asked, for these EQTLs that are correlated with transcription factor levels, are those EQTLs more likely to overlap that transcription factor's binding site? We found that yes, for ChIP-seq peaks, EQTLs that are correlated with a TF are more likely to fall into that transcription factor's ChIP-seq peak. And the same can be said for motif, where if a transcription factor and an EQTL are correlated, we are more likely to see that that EQTL overlaps a motif for that transcription factor. So last, I just wanted to highlight an example that we found. So the LIPA gene likely plays a role in coronary artery disease. The two loci co-localize, and there have been many studies on the LIPA gene and its role in coronary artery disease. We found a specific SNP that has higher affinity for the PU1 transcription factor and is also expressed at higher levels. Across tissues, we found that when there was higher PU1 expression, we saw a larger EQTL effect for this SNP. And in ChIP-seq binding data, we saw that there was allele-specific PU1 binding of this SNP, where there was more binding of the T allele than the C allele. So taken together, we took this information to Dr. Hanhui Zhang and Dr. Feng Li, who have been studying the coronary artery disease locus for some time, and they were excited to look into this SNP and see what role it may have. So far, through luciferase expression assay, they've seen that this SNP does appear to be functional, and that when we have the alternative T allele, we see higher expression. So this is very exciting that we may have found a transcription factor regulator that might be affecting both gene expression and disease risk. So thank you for listening, and thank you to everyone who has helped with this work.
Hello, my name is Giacomo Cavalli, and I'd like to discuss with you today 3D genome organization. And this was a beautiful collaboration with the labs of Elfège Nora and Benoit Bruno in San Francisco. The genome is organized in a hierarchical manner, such that the highest scale of the whole chromosome, you form chromosome territories in the nucleus. At the lowest scale, on the other hand, DNA wraps around histones to form nucleosomes. However, between these two scales, we have not much knowledge about the structures that are formed. And this is a pity, because that's the scale where genes are regulated. With IC, we learned a good deal about it. And so now we know that there are chromatin loops that are formed that join promoters with enhancers or CTCF binding sites. If we zoom out a little bit at the megabase scale, we have domains that have been called topologically associated domains, or TADs, where you have high frequencies of intra-domain interactions, and then they are spaced by CTCF binding sites, and cohesion is binding there too, and so that adjacent TADs are spaced. And at an even higher scale, we have interaction between domains that form repressive or active chromosome compartments. But these are all studies done at the cell ensemble scale, and we wanted to learn about what happens in single cells. And so we developed a technology of oligopaint-dependent DNA fish to study this. And if we now take a look at probes that are within the same TAD in different colors, we see that they frequently intermingle, as you see here. Instead, if we take probes that are painting adjacent TADs, they are much more separated, as you see here, and this is quantified in this scheme. On the other hand, I also want you to pay attention to the fact that TADs are very heterogeneous in shape, and I will come back to this. So this is not just true for a single region. We looked at 15 different regions, and we always see that there is much more intermingling for probes within TADs than between TADs. And this is even exacerbated if cells are led to differentiate into neural progenitors. As you see here, the difference is increased, and this is also quantifiable. What about the determinants of TAD formation? I told you about CTCF and cohesin. So we depleted both of them. And when we deplete CTCF, and we look at probes that span adjacent TADs, we see that they now start to intermingle much more, as shown here and as shown here in the quantification. Such that if you look at all the 15 regions, in fact, you're losing the difference between what you see in probes between TADs or within TADs. They are all looking the same. Basically, the boundaries are abolished. If you now look at the cohesin, on the other hand, and you deplete cohesin, now if you look at the same probes that are between TADs, they are not intermingling like they were doing with CTCF. Instead, the difference is seen for probes within the same TAD that normally intermingle, but they intermingle much less when you deplete cohesin. And this leads us to think that basically cohesin is normally generating contacts within TADs. And so when you lose it, you lose this intermingling within TADs. Now the outcome, the net outcome is that you lose the difference between, between and within TADs, like for CTCF, but for a fundamentally different reason. In the case of cohesin, you normally generate contacts that are not generated anymore, such that within or between doesn't make a difference anymore. Now, if I told you that looking at TADs, we realized they are heterogeneous objects. And actually, we could segment this heterogeneity, and we identified subdomains that we call chromatin nanodomains that have a size of about 200 nanometers and 100 kb. And if we treat the cells with an inhibitor of histone deacetylases, like TSA, we increase the number of objects as quantified here. And this suggests to us that basically preventing histone interactions by hyperacetylating histones breaks down the C and Ds into smaller objects. And so we think that this is an important observation. And to recapitulate, TADs are heterogeneous and subdivided into smaller domains that we call C and Ds that are regulated by chromosome interactions. Within TADs, cohesin promotes long-range chromatin contacts that might be important for enhancer promoter interactions. And CTCF may segregate TADs by preventing cohesin to cross TAD boundaries. Thank you very much for listening.
Hello everyone, my name is Pablo Villegas and I'm here to present this project that I did with Beatrice Borsari as a co-author, and that is based on the study of the role of enhancer-like signatures in the control of tissue-specific functions and homeostasis in human tissues, also considering the genomic location of these enhancers and the degree of specialization of these tissues. So our starting point is, of course, the ENCODE catalog of epigenetic signatures based on the DNA's hypersensitive sites, which I'm sure you all know this workflow that defines almost 1 million enhancer-like signatures across multiple human tissues. So this is our starting point. So by looking at the genomic location of these enhancers, we could see that there is a direct correlation between the percentage of intronic ELSs and the number of samples in human tissues. This means that the genomic location is an important factor in the regulatory role of these enhancers. Since we are interested in the tissue-specific activation of these enhancers, we assess the presence-absence of these enhancers across a curated set of 25 different samples. By multidimensional scaling, we could identify 5 different clusters of tissue-related samples, and this classification is also seen in this heat map that shows the percentage of enhancers that are shared across all the tissues or the samples that we are considering. And here you can see the highest sharing in the 5 different groups of tissues that we have, which are iPS, blood, brain, fibromyoblast, and muscle. In order to consider also these the enhancers that are activated in all the tissues at the same time, we created also the category common. In this bar plot, I'm showing you the genomic location of these enhancers across all the tissues that we're considering and into different genomic locations, intronic, in introns and intergenic regions. As you can see, the more specialized is the tissue, the higher is the percentage of intronic enhancers that they present. On the other hand, the less specialized tissues present an enrichment of these enhancers in intergenic regions. So in order to study more in depth the biological function of these enhancers in terms of gene targeting, we made use of the significant eQTLs reported by the EN-TEx project. So we just, by looking at the eQTLs that are found within the coordinates of our tissue-specific enhancers, we could see the target genes of these eQTLs that we use as a proxy of the target genes of our enhancers. So in this heat map, I'm showing you the percentage of enhancers that present an eQTL within their coordinates. And by looking at their target genes, we could see that they are enriched in the tissue in processes that are related to their tissue. In this case, in brain, they are enriched in brain-related processes. On the other hand, in intergenic enhancers, there's no enrichment at all. We wanted also to see if this specific function of intronic enhancer and tissue-specific enhancers are due to an underlying pattern of transcription factor binding. By using the software Homer, we could see that there is no difference between the transcription factor that bind to the intergenic and intronic enhancers. So this function must be more related to the genomic location of these enhancers. Also, we wanted to know how the embryonic enhancer vary to their development to the adult stages. So by taking 27 different embryonic samples, we studied the genomic distribution, which is similar to the adult tissues, but with less remarkable differences across the tissues. And also, in order to see if this transition between the embryonic enhancer to the adult stages, we checked the sharing of these embryonic enhancers in adult tissues. As you can see, the less differentiated tissues, stem cells and neural progenitors, present a very low sharing of enhancers in adult samples, and the higher sharing is present in differentiated tissues and in the common enhancers, which regulate housekeeping genes. So these are the take-home messages of this project. Basically, the entronic enhancers regulate the expression of genes that are specifically related to the tissue function. Also, there's no underlying pattern of transcription factor binding in the intronic enhancers, and also the genomic location of these enhancers shift towards intronic localization during the tissue maturation. So that's all. Thank you very much.
Hi, my name is Grigory and I want to present to your attention our work, RNA-Chrom, database genome-wide RNA-chromatin interactions. A considerable number of non-coding RNAs are known. Long non-coding, micro, small interfering, PV interacting, enhancer RNAs, and others. They have different cell localization and play an important role in almost all processes of cell activity, including transcriptional regulation, chromatin remodeling, and chromatin architecture maintenance. For the present, various techniques have been developed to localize specific RNAs on chromatin. These include chromatin isolation by RNA purification, capture hybridization analysis of RNA targets, and others. We classified these techniques as one versus all. One specific RNA versus all loci of the gene. In 2017, works began to appear that offered methods to obtain genome-wide data on all potential RNA-chromatin interactions in a cell. For example, mapping RNA-genome interactions, capture of global RNA interactions with DNA, and others. We classified these genome-wide techniques as all versus all. So, the aims of this work are: accumulate all available data on RNA-chromatin interactions in our database, preprocess these data using a single protocol, develop a functionality for the biological data analysis, develop a web interface to make it easier for scientists to use the analytical capabilities of this resource. At the moment, our database contains 34 datasets. These are human and mouse, all versus all data. It is interactions of all RNAs with chromatin, and one versus all data. It is interactions of specific RNAs with chromatin, for example, XIS or MALAT1 RNA. How does our service work? First of all, the user have to select a model organism, then the experiment or experiments of interest to him. Then select the RNA and contacts filter. For example, select link RNA that contacts in upstream regions of genes. After that, create a track of contacts, which will be displayed in the track list with a full description. The main object of this resource is contacts tracks. Therefore, after creating them, the user can perform one of the possible analyses with them. Plot graphical summary. Get a RNA list with all RNAs and the number of their contacts with chromatin. Send RNA-chromatin contacts profile to genome browser. Get a table with the description of RNAs by their types, which were detected in the created track. Download the track for own analysis. This slide shows some examples. Full experiment metadata, list of RNAs that interact with chromatin in this experiment. This table contains RNA loci, observed number of contacts, normalized contacts counts, and other important information that allows judging about the significance of the RNA. Contacts profiles of selected RNAs from different experiments. XIS RNA presented here. Contacts profiles in the genome browser and histone modifications datasets. We thanks Alexandra Golitsina and Razin's laboratory, especially Alexey Gavrilov, for participating in discussions on our project. Thank you for attention.